Welcome to the Rocky Mountain MS Center's webinar series. This series is designed to give people with MS information to empower them to make the best choices for their lifelong brain health. I would like to thank the pharmaceutical companies who have provided support for this program. Thank you to Biogen, EMD Serono, Genentech, and Sanofi Genzyme. Today's topic is a discussion on clinical research in MS with Dr. Timothy Vollmer. Dr. Vollmer is a graduate of Stanford University School of Medicine and a former faculty member at the Yale School of Medicine. He has served as Director of Neurology Clinical Research at the University of Colorado Anschutz campus, the Medical Director of the Rocky Mountain MS Center, and Co-Director of the Rocky Mountain MS Center at University of Colorado. Dr. Vollmer retired at the end of 2021, but continues to be an active advocate, author, and speaker in the field of MS research. This webinar features a lot of important information. If you missed something on a slide or want to hear it again, we are recording this webinar and it will be archived on our website, www.mscenter.org, so you can replay it at any time. All live viewers will get the recording automatically sent to them. You will also receive a link to our evaluation survey. Please take a few minutes to fill this out to help us improve future programming. We'll reserve time at the end to answer questions from the audience. You can submit questions at any time by typing them into the Q&A box on your screen. We will answer as many questions as time allows. And today's presentation is for informational purposes only. All decisions regarding MS treatment and medications should be discussed with your neurologist. With that, I'll turn it over to Dr. Vollmer.
Thank you, Kelsey. Let me share my screen and pull up the slides here. Oops, sharing screen has been disabled by you guys. So while they're letting me share my screen here, I'd like to thank you all for taking the time to join us today. This is meant to be a more informal discussion, so I'm happy to take questions while I'm talking and also go in a different direction if you would like. So do feel free to submit questions through the chat function. Sorry about that, folks. I'm technically challenged. So anyways, as I said, today it's meant to be more of a discussion. And what I would like to do is to start out just by using a few slides to sort of scope out the breadth of research that would fall under clinical research and to give you some ideas of things that are going on. I will not be able to review every clinical study that's going on, and I'll tell you why in a second. So first thing is, what is clinical research? Well, clinical research can be many different things. The point of clinical research is that it's aimed at understanding disease and possibly developing new treatments or diagnostics for disease. So that can be done using in vitro techniques where you grow cell cultures. You can use animal model research. You can use human studies of the biology of MS, genetic studies, pathological studies, imaging studies, et cetera. So there's a lot of different things that can be done. And this is not all the different categories of clinical research. But this list does include studies that we are involved in. So at the Rocky Mountain MS Center Anschutz campus, we have 50 active clinical research projects, and they cover the gamut of the things that I've listed here. From the FDA point of view, though, they think about clinical research in a different way. They think about the stage of the research in terms of its development as for developing a therapy or a diagnostic. So the first category are called preclinical studies. Those are all of the animal studies and in vitro studies and chemical studies that were done to develop the agent under study, whether it's a diagnostic or whether it's a therapeutic. Phase 1 studies are first in man or woman studies. So these are the first time the drug's going to be given to human beings. These tend to be small, very intense studies, and their primary goal is to determine safety and tolerability of the drug. In other words, is the drug safe enough and tolerable enough to go on with larger groups of patients? And it's also used to find the right dose to study. These are complicated studies to do because the number of people participating is relatively small. So the chance to have large variations in outcomes that confuse the issues is quite common. A phase 2 study is when you've completed the phase 1 and you have a dose that you want to test and you didn't find any major safety issues. Then you go to phase 2, which is a larger study, and it's still focused on safety, but now you're going to include a measure of effectiveness of the drug. So you'll be looking at some marker that you identify as a marker that if it improves in the direction you would like it to go, that would be evidence that the drug is actually helpful. So these studies are designed to have a single primary outcome measure, and that's the study lives or dies on that by the FDA standpoint. So you pick out one thing that you think is important, and you try to make that one thing better in a statistically significant way. And if you don't reach that statistical significance, the study is considered a failure. So these studies tend to have 50 to 120 patients in them in the field of MS, and they tend to last 1 to 2 years, take about 4 to 5 years to execute. Phase 3 studies are where you've completed the phase 2, you had positive results on finding the key outcome you were looking for, and now you're going to study the drug in a significantly larger population. These are usually multi-center studies, multinational studies done around the world. And in the field of MS, these usually have about 1,000 patients per arm, 500 to 1,000 patients per arm, and tend to run over 2 to 4 years. And then they take about 6 to 7 years to actually complete. And again, these studies, you have to pick out one outcome that you're going to live and die on. And that could be decrease in relapse rate in MS, or it could be an improvement in function, or it could be a decrease in the rate of disability progression. But whatever you pick out, that first one you have to succeed on, or the study is considered a failure. If you succeed on this and you do at least 2 studies or a very large single study, then you can actually go to the FDA and seek approval of your drug for marketing. And once it's approved for marketing, you're still going to be doing studies in general, and these are called phase 4 studies. And these are meant to find out additional information that would help physicians and patients using these agents, additional safety studies, dosing studies, other outcome studies, trying to demonstrate what the drugs do in a more complete way than you can do in a formal phase 3 trial. So that's when I'm going to show you studies that are going on in this field. Phase 3 trials are studies that could lead to a new drug within the next couple of years. Phase 2s are probably 5 to 7 years away from FDA approval, and phase 1s are probably a decade away from FDA approval. But there's also the type of study design that you're using. And the gold standard, as you probably heard, is a parallel arm randomized placebo or active comparator clinical trial. This is a very special kind of trial. This is where usually both the physicians that are involved in the study and the patients are blinded to treatment. The treatment is assigned randomly by some kind of statistical protocol. Often there's only 2 arms to the active drug you're studying, and then either a placebo arm or an active comparator arm. An active comparator is some other drug that's already approved, that's considered a standard drug in the field, and you're trying to match it or improve upon it. In this day and age, we don't have enough therapies in progressive MS to do active comparator trials very much, so that they tend to be placebo-controlled trials. But in relapsing remitting MS, it's ethically difficult to do a placebo-controlled trial because we have so many good therapies. So those are usually for relapse remitting patients, those are going to usually be active comparator trials. But there can be more than 2 arms. You can actually test 3 or 4 drugs at one time. There's Bayesian designs and other things that you can do. And these studies, again, are complicated, and they have to be evaluated by statistically accepted techniques. And again, the study lives or dies based on those statistics. But even though this is a very powerful study design, because by randomizing and blinding everybody, you're controlling for a lot of variables that could introduce bias into the study. Not every variable, but a lot of them. But it turns out that if you actually compare the results of open label randomized studies, or even open label observational trials, they tend to get it right about 90% of the time. So we don't always need to do a placebo-controlled trial to learn. The issue is that from an FDA approval standpoint, they're going to be worried about biases that gave you the result that you got that actually means that your observation is not true. And so it's harder to convince the FDA to approve a drug in this situation, but it can be done. And then also what we do a lot of in terms of clinical research is chart reviews, where we have about 4,000, 5,000 patients in our clinic, and we tend to treat in a fairly standard way. So we can actually go back and pull charts on patients and look at how they did on the different therapies that we used. And we do collect a number of outcome measures. And chart reviews is also a powerful way to generate evidence for a question that then informs the study design. So it's not going to lead to FDA approval, but it does help inform our treatment patterns, and it does help us design better studies to do. So with that, now I'd like to just do a couple of slides just to briefly review the biology of MS and focus on the different points in the biology of MS, these different treatments we're going to talk about might work. So the first thing is just thinking about the key cells that drive MS. These cells are our lymphocytes, T lymphocytes and B lymphocytes, and they get to the brain by circulating in the blood, moving into the blood vessels in the brain, crossing the blood-brain barrier, where they then get activated and lead to disease. So that in this cartoon, it doesn't work here. Anyways, on the left-hand side, you can see sort of a red line with a gray line underneath it. That represents the blood vessel. And so the immune cells are in the blood vessels and they have to cross that blood-brain barrier. And that's actually a very powerful target for therapy. In fact, the interferons, which were the first approved drugs, work by inhibiting the ability of lymphocytes to cross the blood-brain barrier. And the anti-CD20 monoclonal antibodies, which include Ocrevus and Kcentra and rituximab, they work by killing the cells in the blood so they can't cross the blood-brain barrier. So that's a very good therapeutic target. Natalizumab is the most elegant drug in this class because it simply blocks the ability of the lymphocytes to stick to the blood vessel wall so they can't cross the blood-brain barrier. The next step is that once the cells are inside, they get activated. And that is a key target for many of the therapeutics that are currently in development. Copaxone may work at this level where they inhibit this activation of the T cell and the B cell once they get inside the brain. And some of the newer drugs I'm going to tell you about, like the BTK inhibitors, work at that level. The next level are astrocytes. Now, astrocytes are a key player, and I'll come back to them briefly. Unfortunately, they are not often considered a therapeutic target by the field today, which is a real loss, because astrocytes are one of the three key cells in the brain. The other one's the neurons and the oligodendrocytes, which make the myelin. And we tend to focus on the oligodendrocytes and the neurons, but the reality is that the astrocytes make the brain an environment where those cells can live. And they also regulate the ability of those cells to repair themselves and to survive. So it turns out in MS, astrocytes, which are normally the protectors of oligos and neurons, become actually part of the attack on those cells because they become pro-inflammatory. And we actually have a class of drugs, which are the Gilenya type drugs, and they work by inhibiting these astrocytes from being activated. And that is probably where the BTK inhibitors work as well. And I'll come back to those. And then finally, the other area therapeutically you could target is actually the neuron itself, because it's the loss of neurons that cause disability, or the oligodendrocytes, or both. Those would be the reparative or remyelinating therapies or the neuroprotective therapies. And we don't really have any currently approved, but this is where the major advances are being made in research today. And I'm going to tell you a number of different studies that are focused on this particular area. This is very exciting. First time in history, we've actually been targeting this class with so many agents, that many of which have a very good chance to be useful. Now, just back to the astrocyte. The astrocyte, even though it's a structural cell in the brain, in fact, it makes up more than half the volume of the brain, can either be a neuroprotective cell or it can be a member of the immune system. And in multiple sclerosis, it becomes a member of the immune system and is part of the immune attack on the brain. And this cell should be a major target for companies trying to develop therapies, because by manipulating this cell, you not only can inhibit the inflammation, which is probably drives the progressive phase of the disease, but you can also use these cells to stimulate repair of neurons and oligos. So this cell would be a key target to actually repair the brain, as well as help decrease the inflammation that drives progressive forms of the disease. So let's review a few. The first agents I'm going to talk about are the immunological therapies. This is just a list. And looking at the top here is bone marrow transplant or hematopoietic stem cell transplant for bone marrow replacement. This has been around forever. I think it's using a sledgehammer to try to fix a watch. It's not a very elegant therapy at all. It does decrease inflammation in the brain. How long it decreases is not clear. There's no evidence that it's more effective than the other therapies that we already have, but there is a large study going on in the United States to compare this to the best of the disease-modifying therapies that are approved by the FDA. The next group are a very interesting group of therapies because these therapies are meant to be curative. These therapies are aiming to try to remove whatever's driving the disease forward. So the first one is a cell line that's grown in culture. And the cell line is a type of mesenchymal stem cell that's involved in repairing the blood-brain barrier. And so they're injecting these intravenously to try to repair the blood-brain barrier, the hypothesis being that the problem in MS is the blood-brain barrier is defective and allows immune cells to get into the brain. I think it's a long shot, but it is an elegant approach. The next one is another cell line. This is another human cells that are grown in vitro. These are T lymphocytes or T cells, and they target the Epstein-Barr virus. And so this is being tested on the hypothesis that Epstein-Barr virus actually drives the disease. And that hypothesis has been around forever, but this would be a direct test of it. Again, a fairly elegant approach. It's basically giving people immune cells to help clear Epstein-Barr virus infected cells in hopes of then shutting down the inflammatory disease process in MS. The next two therapies, Neurovax and this myelin glycosylated binding to Kupffer cells, they are meant to reprogram the immune system. So they're to try to develop tolerance. And the Neurovax is a mixture of peptides from T lymphocytes that target myelin basic protein. And these are, again, T cell receptors that have been genetically engineered and isolated and create the peptides. And they made a vaccine out of them, and those are being injected into patients. The hypothesis being that by injecting these proteins from the T cell receptors, that will lead to an immune response against them, which will then kill the immune cells that are actually driving MS, the assumption being that they share these T cell receptor patterns. The next one down is actually a very interesting one. This is where they're actually taking myelin peptides and adding sugar molecules to them that specifically bind to an immune cell in the liver called a Kupffer cell. And the Kupffer cells are very important because they routinely screen your blood for any kind of foreign protein or other molecules. And they internalize them and then process them and re-expose them on the surface and show them to the immune system. And they do that in a way that they educate the immune system about what's out there. And they can either tell the immune system to attack it or to protect it. In this case, they're trying to use the Kupffer cells to tell the immune system to protect cells that have this particular protein, which is myelin basic protein. And then the tenofovir, this is actually a drug used in HIV, and it's being studied in MS because there's a hypothesis that there are genes that have been incorporated into the human genome that are called these human endogenous retroviruses. So human endogenous retroviruses are a lot of otherwise useless genetic material that surrounds other genes in our genome. And they were incorporated into our genome 100 million years ago. And they've been just accumulating, millennia after millennia. And normally they don't do anything, but there is some evidence that they can contribute to some diseases. And there's a hypothesis that protein products from these retroviral genes that are built into our genome are actually driving MS. And so the hypothesis here is that tenofovir is an antiretroviral agent, can inhibit the body's ability to reproduce these genes and thereby decrease the production of the proteins that are related to these viruses to see whether that actually might have a positive effect in MS. And then the next one down is a drug we don't know too much about. The company hasn't really published much about it, but it's meant to clear B lymphocytes from the brain of MS patients. We know that B cells are critical to the disease process. And this is a molecule, it's a small molecule. It's a drug that inhibits the ability of plasma cells to work. I'll come back to the BTK inhibitors, Brutus tyrosine kinase inhibitors. These are the major ones likely to get approved in the next few years. And then just finish up with these last four. Last four are monoclonal antibodies, very much like ocrelizumab or rituximab or natalizumab, but these target different molecules. And they're targeting very specific regulatory molecules that are involved in the regulation of inflammation in the brain. Monoclonal antibodies are wonderful molecules because they're extremely targeted. You can target one cell out of a trillion with these molecules and with no bystander injury. So these are very powerful approaches to therapies, and these are new ones that are in clinical trials or about to enter clinical trials. Just a little bit more on the antibodies. Antibodies are becoming one of the most common medications we use in medicine today for many different reasons. And there are basically 4 different versions from the FDA's point of view. There are, on the left-hand side, it says the murine, which means mouse antibodies, and they're named by having MAB added to the end of whatever name they give them. We don't really use any of those in MS anymore. The next one are the chimerics. Rituximab is a chimeric. It's 2/3 mouse sequences with 1/3 being human. And that was the best they could do 30 years ago when they first built that molecule, and it's a very good molecule. The next one over are the more humanized ones. These are called Zumabs, so ocrelizumab, natalizumab, alemtuzumab. These are more humanized monoclonal antibodies, and that ZUMAB on the end tells you that they're humanized. And then the fully humanized ones are just coming out, and these are the ofatumumab, opisumab, and these are molecules that are fully human. They don't have any mouse sequences in them, and they also can change the sugar moieties. It's called glycosylation. They can change the sugar moieties that are attached to these molecules, which changes their behavior. And this is the real future. These are easy to generate now, and they will be replacing some of the older antibodies as we go forward. And then the BTK inhibitors. As I mentioned, these are oral medications. They're small molecules, and they inhibit an enzyme called Brutus tyrosine kinase. And that molecule is a key regulatory molecule. There are 5 that are fairly far along in development. There are at least 2 others that are in early phase development. We have preliminary data on phase 2 studies from 3 of them, and they're all in phase 3 trials. And the data so far suggests that they're going to be roughly as effective as Gilenya in that class of drugs. Their safety profile looks like it's probably going to be better, and probably going to be pretty tolerable drugs, not a whole lot of side effects. So I think from the standpoint of oral drugs, this probably will be become one of the more dominant used therapies if they get approved. So we have to wait until their data is submitted. The major challenge for these drugs is actually the development process. It turns out that the Russian assault on Ukraine has severely damaged these studies because that part of the world doesn't have as much access to modern medications as United States, Canada, and Europe does. And as a result, there's more patients there that could use these kinds of therapies. So the companies had been doing these studies in those countries. And unfortunately, because of the war, on both the Russian side and the Ukrainian side, many of these studies have stopped and the patients have been lost. That may end up making these studies not able to succeed because the numbers may be too low, in which case they'll have to start over. So this is a major challenge, and we'll have to wait to see how it turns out. But I think that unfortunately, it's probably going to slow the development of these by at least a couple of years. And then this is just different signaling pathways where the Brutus tyrosine kinase is actually used. The one on the left is the one that most people hypothesize would be useful for MS because it's involving B cells. But the other 2 on the right-hand side are active in many other cells, including astrocytes. And I do have reasons to believe that these drugs may be working primarily through their effect on astrocytes because they have a much bigger effect there than they do on B cells. But nevertheless, they're being developed based on their effect on B cells. However, if they do get approved, we do expect to launch phase 4 studies because these drugs might help induce repair. And that would make them special if they actually were able to induce repair. And then finally, these are studies that are going on in the neuroprotection or neural repair area. I can't tell you about all the studies because there's 2,614 clinical trials listed in clinicaltrials.gov on MS alone. And so, but anyways, I pulled these out as sort of examples. Again, there's the stem cell transplant, which I don't think is likely to work. Ibutylast is a neuroprotective agent. It's approved for use in Japan. And it did a phase 2 study and showed that it did preserve brain volume. And preserving brain volume tends to predict improvement in disability scores. Clemastine is an antihistamine, also in early phase trials because it's a neuroprotective agent. Metformin is medication used for people who have prediabetes or diabetes, but it also enters the brain and it induces neuroprotection, primarily through astrocytes. And then the EK12 is a mixture of drugs that activate endogenous opioid receptors. And that's because those receptors are targeted in pain, but they're also potentially neuroprotective. EHP-101 is a cannabis product. It's a cannabidiol, and it's because the cannabis products activate a group of receptors in the brain. These cannabinoid receptors are involved in many different things, but they also are involved in neuroprotection and repair. There's a small molecule, the BIIBO61, induces oligodendrocytes to divide and make more myelin. The lozanimab is an anti-repulsive guidance molecule. There was a question about that in the early questions. This is a monoclonal antibody that helps to block a molecule that's in the MS scar that prevents neurons and oligodendrocytes from putting out new branches, similar to the anti-lingo studies that were done by Bajin before. There is a study going on with testosterone and neural protection. I'm not quite sure what the hypothesis is there. And then there's studies looking at new drugs for spasticity. The biximols are the FDA name for the cannabis, cannabinoids, and THC. And it's a 50-50 mixture of THC from marijuana with other cannabinoids. And then the next one down is also activating cannabinoid receptors. So these are things that are being spun off from the marijuana field, but they have a very strong science behind them. And actually, nabiximols are already approved in Canada and England for spasticity. This is another list that I included in the slide set, and we can make these slides available to you. So if you're interested in a particular agent here, it lists the companies here. So you just have to do a search on the companies or go to clinicaltrials.gov, and I'll give you that website in a second. And that will tell you where the studies are being done, and you can contact them to follow up with them. And the last drug I wanted to talk about was semiglutide. This is a drug that we're going to be working on and hopefully can get funded. This is another drug that's actually used to treat diabetes and prediabetes, but it acts by activating a hormone called glucagon, which is produced in the brainstem. And it also turns off those pro-inflammatory astrocytes and shifts them in a neuroprotection direction. The other nice thing about this is that part of the cause of disability in MS is actually having other diseases like high blood pressure, diabetes, elevated cholesterol, cardiac disease. And this is the most effective drug at eliminating those diseases that we've ever seen. So it causes people to lose weight because it suppresses appetite and delays gastric emptying, which makes you feel fuller longer, so you don't eat as much. And it was able to normalize hemoglobin A1Cs in diabetics, prediabetes patients at 70% level over 2 years. It normalized blood pressure in 60 to 70% of patients. And doing that helps the brain repair itself and recover function. So not only may it be a neuroprotective agent that might help the brain repair itself, it also may remove some of the other stresses on the brain that increases the disability from MS. It's a very easy drug to use. It's a once-a-week sub-Q injection you do yourself, a very tiny needle. And the side effects are really minimal. It's actually one of the safer drugs that we have. So anyways, we're going to try it initially to focus on decreasing the comorbid conditions of high blood pressure and prediabetes, but ultimately to look directly at neuroprotection by looking at biomarkers of neural injury, particularly neurofilament light and GFAP. So having said that, we have made a lot of advances. So we've talked about this concept of brain health, maximizing brain health over a lifetime in MS patients. And we do know a lot of things that really do help them. So using highly effective therapies as early in the disease course as you can, exercising. Exercise is then repeatedly demonstrated as being the most effective way to improve symptoms in MS. We've had hundreds of studies. Staying active and learning and art, having healthy leisure time activity, socially active, have a healthy diet, and obviously treating any comorbidities like high blood pressure, diabetes, or obesity. And if you want to sort of get more information about any of these studies, there are some good sources. Clinicaltrials.gov is run by the FDA and the NIH. And it's a website that lists essentially all of the clinical trials in North America. And so it's a good place to go find out. It doesn't really tell you why they're doing the study, but it tells you how the study is designed and who's doing it and where the study is at in terms of if it's enrolling or not. And it usually gives you contact information if you want to contact them. PubMed.gov is a literature search engine run by the government. It acts as the medical databases. And so it gives you actual medical articles that you can look up. It's a very useful tool. You'll mostly get abstracts, but that's where the meat of the paper is. So you only have to read a paragraph, find out what the study was about and what the key results were. So you don't have to read a lot of stuff. And it does have some full text articles. But if you want to get the full text of an article, you may have to use Google Scholar. And Google Scholar actually is very good at pulling up medical literature. So that's one. And then, of course, our organization, our website is mscenter.org. We have a lot of information about research, National MS Society, and other organizations do as well. So to conclude here, this is the list of the currently approved MS therapies. There's 23 of them. Ublituximab, which is under the infused medications, is not yet approved, but it is being reviewed by the FDA and could be approved anytime. And that would make 24. And so, remember, I started in this field and finished my training in 1987. At that time, we had no treatments. And it was a very unfortunate situation. We had very little offer patients at that time. First drug was approved in 1991, and it's obviously exploded beyond that. So there's 24 drugs on this list. Having said that, though, it only represents 6 actual classes of medications. So most of the drugs on this list are copycats of drugs, other drugs. For example, there are 5 interferons: Avenix, Beta, Seron, Extavia, Plegridy, and Rebif. There are 3 Copaxone type drugs: Copaxone, Glutamrastate, Glatopa. And the only thing that's in the injectables that's really new is Kcentra, which is really should be over here. And they're all medications. We actually only have 3 types of medications here. We have 2 chemotherapies, Albagio and Mavenclad, not very good drugs in my view. We have 4 fumarates. That includes bafratem, dimethylfumarate, which is Tecfidera, and Vumerity. And then we have 4 S1P receptors antagonists, which are the mods, the fingolimod, siponimod, ponesimod, ozanimod, which is Gilenya, Mayzent, Ponvoy, and Siposia. And over here, Novantrone is an old chemotherapy drug. There's really no role for it in MS anymore. It's way too toxic. Lemtrada is being withdrawn from the market, so it's not really relevant to us. And all of the other ones that are infusable over here are either Tisabri or the anti-CD20s. We have ocrelizumab, we'll soon have ublituximab. There's rituximab, which is not approved, but it's the same drug. And then we have Kcentra, which is the other anti-CD20s. We have 4 anti-CD20s. So even though there's a lot of drugs approved, it's only 6 classes, and 2 of the classes, or 3 of the classes are not very effective or not very safe. So that means we only have 3 classes of drugs that really are pretty safe and pretty effective. So developing these new therapies is really important for us. We need more options. So today, we can discuss more anti-inflammatory or immunological therapies. We can talk a little bit more about remyelinating and comparator therapies. And we can certainly talk about the clinical research going on at the Rocky Mountain MS Center. So let me get out of this. And do we have questions, Kelsey?
We do have a couple questions. The first one that came in is on the BTK inhibitors, covalent versus non-covalent. What's the significance?
Okay, so covalent means that it binds to the enzyme by actually sharing electrons. So it actually joins the 2 molecules together so they become one molecule. That means they don't fall apart. And so the only way that enzyme can become functional again is for the cell to make new copies of the enzyme, because whatever's around when you took the drug have been neutralized. Non-covalent binding means that it's binding more like a magnet, and it can dissociate. And so the drug can fall off of the enzyme, allowing the enzyme to become more active again. And the reason that a couple of companies went the non-covalent route was because if you get into trouble, theoretically, you could clear the drug out of your system faster to get out of trouble. Outside of that, it doesn't have any particular implications.
On Semiglutide, would that be an add-on therapy?
I'm sorry, which one?
The weight loss one?
Oh, semaglutide, yes. It would be an add-on, probably to one of the monoclonal antibodies would be probably the best, just because it limits other interactions. The monoclonal antibodies are really nice because they're so targeted. They have very little in the way of off-target effects. And so combining them with other drugs is technically easier because you can monitor the potential interactions much more easily than you can with something like Gilenya or Tecfidera, which has a lot of effects on all sorts of cells in many different ways. It gets very complicated. But that would be my plan. And that's the studies we're designing, is to add it on to one of the intravenous infusion drugs or Kcentra.
Why is Lemtrada being withdrawn?
The reasons are mainly two categories. One is the development of other autoimmune diseases. So Lemtrada, it turns out, does suppress the inflammation that drives MS, but it does it by changing the way the immune system regulates itself, and that releases the immune system to cause other autoimmune diseases, most common of which is thyroid disease, but it can actually cause many different kinds of autoimmune disease, including autoimmune encephalitis and meningitis and other very serious complications. The other one is serious infections in cancer, because again, you're suppressing the immune system, so that releases infections in cancer.
What are the exercise repair studies?
So the exercise studies have not been focused on repair. They've been focused on whether people recovered function. However, the way that you recover function is by rewiring the brain. So remyelination is generally discussed as a reparative therapy strategy in MS, but the reality is it's not a very powerful mechanism. The more powerful mechanism for recovering function in MS and any other neurological disease is to rewire the brain, to increase the number of connections between the nerve cells you have. That's how the brain basically recovers function. So if you have a significant brain injury or you have a MS attack, you get better because your brain reorganizes and reconnects in new ways to get around whatever's blocked. That's neuroplasticity, and that can be stimulated in a number of different ways. And some of the therapies that I mentioned in those earlier slides are actually targeting that particular issue, even if the investigators may not realize that. So that's why exercise is also would be under the neuroprotective, neurorepair concept. In my view, if you're going to do a study of a drug to increase nerve cells' ability to rewire and reconnect, you have to add exercise into that, because you can create an environment where the neurons and the oligodendrocytes can make new branches and connect with other cells they need to connect with. But that connection will only last a short period of time unless it's activated. You have to send an electrical signal to those new neural connections to make them permanent. Otherwise, they break within 24 hours. And so if you don't have an exercise program related to whatever disability you're trying to improve active at the time you're taking the drug, you're not going to capture very many of those random connections, and therefore you're not going to add on to the connectivity that actually makes a difference. So that has been missed by the field so far. So Biogen has probably done the largest studies in repair with their anti-lingo monoclonal antibody, but they made two very serious mistakes in my view. One was they did not use a highly effective anti-inflammatory therapy. They used Avonex or no therapy. And that was under the hypothesis that you have to open the blood-brain barrier for the drug to get in. And so you need inflammation. But of course, if you allow that, then you allow more damage to the brain, which most likely will erase any benefits you got. And the other one was they didn't have a rehabilitation or an exercise program attached to it. So it meant that in the short time frame of the study, it wasn't likely that they were going to be able to detect in a clinically significant, statistically significant way improvements in patients, because patients weren't doing anything to really improve. So exercise is the single most important thing people can do. And we do need to do a lot more exercise research to understand how to tailor that for people to really optimize it. So those studies will continue to go on. And I hope that the field will become more aware of this issue in future studies on neuroprotection and neurorepair and begin to add on to it exercise programs. The semaglutide studies that I mentioned that were done in pre-diabetics and people with hypertension and obesity did have an active exercise and counseling program attached to them. And they showed excellent results. There's now, that's a class of medications. There's going to be several other drugs coming out in that family of drugs that will be targeting specifically weight loss and improvement in general health. So that's one to watch, I would say.
And is the semaglutide being used now, or is it still in trials?
No, semaglutide has been out since 2017 or 2015, somewhere around there. It was approved for people who had obesity or people who had weight-related health problems like high blood pressure or prediabetes. And then it was approved last year, or actually 2020, by the FDA specifically for weight loss at a higher dose, 2.4 milligrams, called Wegovy. And that's been out for a year to 2 years now. The problem with it is the company made it a very expensive drug. It's not approved that much by insurance. You can get it compounded for a much, much lower cost. So it's something that I think will change in the near future because 3 other companies are making similar drugs. They'll be coming out. So hopefully that will generate competition in the market and help make the drugs more available. But it's unfortunate that the company decided to go the route they did because they dramatically limited the penetration of the drug, and obesity caused disease is the leading cause of death in the United States. That's cardiovascular disease and diabetes.
We did have a lot of questions submitted at registration on kind of the differences between remyelination, neuroprotection, neurorepair. Can you just kind of explain the difference between those and kind of some of the avenues that they're being approached?
Right. So remyelination has been the focus for a long time. So myelin is made by oligodendrocytes. And one of the slides I had is the cartoon there of the oligodendrocyte at the bottom wrapping the green myelin around the nerve fiber. And historically, this has been a cause of demyelinating disease. And that's unfortunate because there is demyelination in the MS plaques. But the more important thing was actually the death of neurons and the destruction of axons. That's what actually leads to disability. But nevertheless, the basic science field views MS as a disease of myelin because that's what they were taught in their limited courses in this disease. And so there has been a lot of research on myelin biology. And there's a large program at the University of Colorado. The first efforts to actually try to help the MS plaques remyelinate were stem cell transplants. So I did one when I was at Yale. We did a phase 1 trial there. And we took myelin forming cells from the peripheral nerve of patients, isolated them and injected them into the patient's brain. And they grew, but they didn't migrate, they didn't spread out. And it turned out to be somewhat traumatic to the brain. So I think the mesenchymal stem cell transplant for remyelination is under the same hypothesis that you make more oligodendrocytes, therefore you can make more myelin. The Biogen had a molecule called anti-lingo for many years, but they changed the name before they discontinued it. And it was meant to actually stimulate those oligos to put out new branches and remyelinate. The anti-RGM antibody that was in the list that I showed you, that's what it's supposed to do. It's supposed to remove or block a protein that's in the MS plaque that inhibits the oligodendrocyte precursor cells, the early cells that make oligodendrocytes, from actually maturing into effective oligodendrocytes. So in that situation, you're focusing on the oligodendrocyte. That's your therapeutic target. And my sense is that probably the best you can do with that kind of a therapy is to help decrease the amount of variation people have in function throughout the day and the week. As patients know, their ability to function varies tremendously depending on the time of day, body temperature, dehydration, all those things. And that's because they have a lot of demyelinated axons that are very sensitive to any environmental changes. Myelin shields them from that environmental change. So if you can make more myelin, you would decrease that negative impact of those environmental shifts on the function of the nerve fibers. Other people think they would result in more significant recovery of function. We'll have to wait and see if they're successful. Now, neural repair is basically both remyelination and rewiring. They both fall under neural repair. But rewiring, cortical rewiring or neuroplasticity is actually the more potent way to recover function in almost any situation. And so in my view, that means that what you have to do is you have to get neurons to put out new branches, more frequent branches that connect with other nerve cells. And then you have to have people doing something they want to make better so they can activate those random connections and make them permanent. And by doing that, build that neuronal tree that subserves whatever function you're trying to improve. But the best way to do that is to treat astrocytes, because astrocytes are the ones that stimulate the nerve cells to put out those branches and helps guide them and protect them. So in my view, we already have a lot of drugs that work through the astrocyte. And the astrocyte, I think, is the key driver of the progressive forms of the disease. And other people have similar thoughts, although it's not a consensus yet in the field. I think I'm right, but we'll have to wait and see. But anyways, so in those therapies, you would aim at astrocytes and potentially agents that help protect neurons from dying. So there are calcium channel blockers and other things you could use for that. They have been tried, they haven't worked so far. But nevertheless, those are the options. And then neuroprotection is more just the latter, where you're simply trying to prevent cells in the brain from dying. And that can be astrocytes, which are killed very early in the MS disease process, neurons, and oligodendrocytes. So there you're looking at the products produced by the inflammation that actually lead to cell death, and you try to block the effect of those. And there's a number of cells, a number of drugs in those lists that I showed you that's what they're aiming at, the proteasome inhibitors and those kinds of things.
So you mentioned, oh, go ahead.
We had a question about neurofilament light. So there's a question about neurofilament light treatments. Neurofilament light is actually a molecule that's released by nerve fibers when they're damaged. So it's not a treatment, but it's a way to monitor how much damage is occurring in the brain. And so our goal is to get it approved as a diagnostic, a blood test that we could do that would help us measure how much injury to nerve cells is going on in the brain, both to help diagnose people who have disease like MS, as well as to monitor the effectiveness of treatment. There's another protein that's produced or released by astrocytes when they're damaged. It's called GFAP. And that also shows up in the blood of patients. And we've published this a couple of times now. And it is also a way to monitor what's happening in the nervous system. And you can measure in the blood, so you don't have to do spinal fluid. So these are called biomarkers. And biomarkers are developing pretty rapidly right now because we have technology that allows us to measure one molecule in 10 to the 12th to 10 to the 16th, which is a very small number of molecules. And I think they will help streamline research, particularly early phase research, because if the FDA buys off that improving neurofilament light levels correlates with improving function, then they might allow us to use that in phase 1 and phase 2 as a much more rapid way to screen drugs and get them through those phases and get on to phase 3, which is the meat of the subject. What other questions did you have?
You mentioned that the MABs will be moving to being fully humanized over time. Do you think it's better to be on a fully humanized DMT or something more like Ocrevus or Rituxan?
So it's an interesting question. So the companies are banking on that they will be able to market the drugs because it's fully humanized, and that sounds better. But the actual data about safety of them and efficacy doesn't really show that there's much of an effect. For example, rituximab is 70% mouse protein, about 30% human protein. And yet the major problem with that is you would think that injecting a mouse protein into human bloodstream would cause humans to develop antibodies that would then neutralize the drug. And it does happen, but it's rare, maybe 2 to 4%. And the frequency of similar antibodies in ocrelizumab treated patients, which is a more humanized antibody, is about the same. It's in 2 to 4% range. Whether the fully humanized monoclonal like Kcentra, ofatumumab, whether that will have significantly lower antibodies remains to be determined. We don't know yet. So I don't think there's a major difference between them. I think that companies will move in that direction simply because when they're developing the molecule, they can. It's easy to do that as it is to make a chimeric. So there's no real reason to stick with chimerics.
Why do they use Aubagio so often as an active comparator if it's less effective?
Because it is less effective, so it's easier to beat. It's like stacking the deck in a basketball game. Put all the short people on one team and all the tall people on the other team. So it's an FDA approved drug, and if patients are willing to take the chance, they're going to be on Aubagio. Because many of these studies are double dummy designs. That's not a description of the physicians. That's a description of that that all patients take both therapies, or they do an infusion, which may be a placebo or maybe active drug, and they take a pill, which may be a placebo or an active drug. So they do both things. So it eliminates that bias that if people believe that infusion is always going to be more powerful than a pill, then you could bias your study, and that takes that bias out.
What happened with the trials on simvastatin?
So the trials on simvastatin were done both because it theoretically could have an anti-inflammatory effect, primarily because of its effect on the antigen-presenting cells, and now I would say primarily because it has an effect on astrocytes, which are part of the antigen-presenting cells in the brain, probably the key one. But it also decreases production of nitric oxide. So statins like simvastatin, which is I don't remember the brand name anymore. Atorvastatin is Lipitor, but all the statin drugs help decrease the risk of heart attack because they decrease the production of nitric oxide in the atherosclerotic plaques and arteries. It's the production of nitric oxide that develops that scar that becomes an atherosclerotic plaque that narrows the lumen. And production of nitric oxide in the brain in MS patients, we think, is a key driver of MS. So the theory behind it was based on that. And there were some other things suggesting some neuroprotective effects. And what we showed was that it could decrease MRI disease activity by about 30%, which is about the same as Copaxone does. But we had to use it at a very high dose. We had to use it at 80 milligrams to get enough across the blood-brain barrier. And that led to a lot of muscle pain and muscle weakness. And so it just wasn't really going to work because of the tolerability issues, because of the muscle pain and muscle weakness with it. It was also studied in early phase disease where atorvastatin was, and it was stopped for the same reason. It had some minor benefit, but it wasn't enough to overcome the tolerability issues with it. However, the BTK inhibitors don't have that effect, and they have a similar mechanism of action. So they may be a way to get to that with a more tolerable therapy. And I think Gileni also could be considered in that category, but it has enough problems that the BTK inhibitors right now, in my view, might be superior from a safety and tolerability issue.
Can you speak just a little bit, we only have a few more minutes, but on any research coming out for progressive MS, both secondary and primary?
Actually, if you look back at the list of the slides that I gave you, about half of those agents on the immunomodulating therapies are actually targeted for progressive patients. And if you look at the reparative ones, almost all of those are targeted at progressive patients. So I would say that actually, if you look at the bulk of research right now, the majority of the research in terms of the number of agents that are being developed are actually targeting progressive disease. So there's more than 20 agents out there now focused on progressive disease from many different angles. So this is a very exciting time. Obviously, we've been hoping to get into that position for some time, but it is there. And part of the reason is because our drugs that we have, like the anti-CD20s and natalizumab or Sabri, are so effective in early phase disease that's a tough hurdle for a company to beat these days. But progressive disease is still wide open and still something that companies can compete in.
And we will be sending out the slide list so folks can look those studies up. Mm-hmm. We had a question earlier about what new treatments are available for foot drop, and also is Ampyra still recommended for leg weakness?
Yes, Ampyra is still recommended to help improve gait, and it is moderately effective. So it's something well worth trying if you don't have contraindications to its use, which is primarily seizures and cardiovascular problems. There actually were a number of studies listed in clinicaltrials.gov looking at foot drop, ankle orthotics, exercise programs to improve gait. I would say there's probably 20 studies in that area. There's some of them were using peripheral nerve stimulators. There was some other technology, I don't know what it was, frankly. But yes, there are research projects going on in that area, but I think the peripheral nerve stimulator is probably the farthest along, and they are approved to help with foot drop. You can prescribe them, and the problem is you're just not covered by insurance yet.
So we are running out of time for this evening, but I thought it might be nice to close with a question. You talked about a lot of the different types of trials and studies out there in MS. Can you talk just a little bit about the value of MSers participating in research? And then also, is there a way for family members to participate in research?
Yeah, great question. Actually, participation of patients with MS in the clinical research process is extremely important. It's essential. Obviously, we can't do it without it. But it's not in just being a participant in a clinical trial. It's actually consider joining the research team, helping design the studies. The reason is that we have to pick out outcome assessments. So we might measure ability to walk, we might measure vision, we might measure pain, we might measure speed or balance or falls. But what's important to patients? And so if they don't participate in the process, we may not pick out the right ones. And we actually found that in our work earlier with some of the focus groups that we used, particularly with the patient questionnaires. The patient questionnaires that patients thought were most relevant and most important to their experience were not the ones that we had picked out. So that was very educational for us. The other reason to get involved in clinical research is because there are a lot of decisions about how drugs are going to be administered, how it's going to be monitored, even advocating on cost issues, those kinds of things. So we're involved in sort of that whole process, not just testing an idea in the clinic, but actually figuring out how it fits into the clinical environment. How does it compete with other drugs? Which patient should it be used in? Recommendations to the company and ways to market it that accurately tell what the drug can do, and also constantly advocating for the companies to try to make these drugs more available and more cost effective. So there also are types of studies where participation directly by patients with MS is critical. There are a number of databases, including the NARCONS project that we started years ago, which is still going strong. And those databases are very valuable. So participating in these databases where you add your information to it and then made available to researchers to analyze and ask questions of it really helps tell us what's happening to the population and what are important questions that we need to address with other studies. So it's a way to inform the research enterprise to identify what key issues that we need to move forward on. Also, there are 24 drugs approved for MS right now. There'll be maybe 30 by the end of the next 3 or 4 years. We need to find out which of those drugs really work the best and for whom. That's not going to be done by randomized trials. That's going to be done by patients and clinics working together to standardize the way that we treat so we can use different drugs in a certain way and follow key outcomes like your ability to walk, like ability to be employed, like fatigue, like depression, like employability. And so we need to collect that data as part of the medical record system so that we learn as we go through. So medicine is one of the few businesses, large businesses in the United States that is not a learning organization. And that's an old business term. I don't think they use much anymore, but it came out in the 1990s or so. And the concept is that the company actually monitored their products to figure out what was working, what was not, so they could constantly improve that. Medicine, once you walk out the clinic door, we don't know what happens to you. So we need to really try to have patients stay connected with clinics that are involved in this research. Not going to do primary care there, their primary medical care there, but still work with them to help gather information on how they're doing, what therapies they're on, what problems they had from them, so we can identify benefits and risks and then inform people more accurately and also design further studies to clarify those issues. And then there's a whole lot of other issues. Like, so the question was, can family members participate? Absolutely. There's a lot of work done on genetics. And then we have the RISE and DREAM MS studies. So these are studies trying to identify biomarkers that identify children born into families with MS who are more likely to develop MS themselves later in life. That is a very important study for several reasons. One is that it gives us ability to begin to gain insight into the very earliest immune mechanisms that lead to the disease. But secondly, it may lead us to a way to diagnose those patients much, much earlier before they incur any significant brain injury, so we can treat them before they have to go through MS relapses and disability and all of those things. So that's a study for children over the age, I think it's 10, but you may know better. And then older adults over the age of 18 up to age 35, if I remember correctly. And basically all that's involved, yeah, maybe 30. All that's involved is that you get an assessment where you get some blood drawn, you get an MRI done that we pay for, and you get some questionnaires and some general assessments. In the blood, we store in our biobanks, and we analyze for markers of brain injury as well as immune markers that are related to how the immune system's regulating itself. So that would be a great study for family members to get involved with. And then, as I said, there are studies, centers working on the genetics. And those are the main areas right now. So early diagnosis is a key, and then identifying the early biology, at least MS, is a key goal. But again, we have many types of research, and sometimes it just involves answering a single questionnaire or donating a single sample of blood. Sometimes it's being followed prospectively for 10 years. So there's lots of ways of getting involved.
We did put the information in the chat for our research recruiters at CU if you're interested in any of the studies that they're doing there. But thank you, Dr. Vollmer, for your time tonight. Thank you, everyone, for your great questions. Obviously, research is a huge topic with lots to explore. So I just wanted to remind folks, we will be having Dr. Corboy discussing the updates of the DISCO MS trial on June 13th. You can sign up through our website or Eventbrite for that webinar. And then we also make sure to have research update components to all of our education summits, which the fall education summit will happen on October 15th. We just had the most recent one on April 30th. You can see that video archive on mscenter.org or YouTube. So thank you guys. Make sure you're signed up for our mailing list in order to receive notifications about upcoming education events. And also please do take a moment to fill out the survey and let us know what you'd like to see in the future.
Thank you all.
Thanks, Dr. Vollmer.